All right, hey there. So when we last talked, we had discussed the structure of DNA and DNA replication. Okay, so now we're going to move into how DNA functions. How does it function as a blueprint, quote unquote, for life, right? So this is what we call the central dogma of biology, is how information flows from DNA to RNA to protein, how that information in DNA gets used to build proteins. Okay, so the first step that we're going to talk about in this process from DNA to RNA, that's called transcription. So we take the information in DNA, we're going to make a copy of part of that DNA molecule into an RNA molecule. That's our first step, transcription. So in eukaryotic organisms, DNA is going to be contained inside of the nucleus, and so this whole process of transcription is happening inside the nucleus in eukaryotes, right? So there's pieces in our genome, we have pieces of DNA called genes, and these genes are pieces of DNA that code for a specific protein. Okay, let's say we're going to build the lactase enzyme, for example. There's a segment of DNA on one of our genes, and a segment of DNA called a gene that's going to code for the information for how to build that lactase enzyme. Okay? But in order to get that information out of the gene, we need to make a copy of it. I'm going to make an RNA copy of that gene, of that piece of DNA, because we don't want anything to happen to that original piece of DNA. We don't want any sort of errors or damage, anything like that to happen to that DNA. So we're going to use a copy of it, right? So I use the analogy of, let's say I have a whole bunch of old handwritten recipes from my great-grandmother. They're handwritten, they're on this really old paper that's starting to yellow, and they're really special to me because these are family recipes, right? So I'm not going to take those original old handwritten recipes. I'm not going to take them with me into the kitchen when I make cookies or something with my 3-year-old cousin, right? I'm probably going to take a picture of that original recipe and then take that into my kitchen with my cousin, or maybe make a photocopy of that recipe, right? Because I want to keep the original as pristine as possible. That's the idea behind transcription. We want to keep the original DNA as pristine as possible. We don't want any damage to happen to it. But these copies that we make of it, these RNA copies, these are expendable. These are shorter pieces, and we can make as many copies as we need to. So in transcription, what's going to happen is that we're going to have an enzyme, RNA polymerase, this light pink blob right here. RNA polymerase is an enzyme that builds RNA. So we saw DNA polymerase. DNA polymerase is responsible for DNA replication. RNA polymerase is an enzyme that's going to build RNA molecules. And so what this RNA polymerase is going to do, it's going to be attracted to this region right here that we call the promoter. So this promoter, this is a sequence of DNA. It's usually TA repeated, so it's sometimes called a Tata box. It's a sequence of DNA that RNA polymerase recognizes as a location to start the process of transcription. So RNA polymerase will attach itself to that promoter region, and then it's going to move along the DNA, open up a small little replication bubble of DNA, so it's going to separate the strands, the two pieces of DNA, and it's going to read one strand that we call the template strand, and it's going to find complementary RNA bases to that template strand. So if there's an A in the DNA in the template strand, it would match a U, an RNA U, with that base. Or if there's a G in the DNA template strand, it would match a C, an RNA C, to match with that, right? A cytosine. So it attaches originally, RNA polymerase is going to attach originally to the promoter, and then it's going to be moving along that template strand and building that single-stranded RNA copy. So here's another example of what it looks like. So it's going to keep moving along that piece of that gene. Okay, it's going to move from that promoter region. It's going to move towards the 5' direction. Okay, of the original DNA strand. It's going to move towards the 5' direction, and it's going to continuously add on new RNA bases that are complementary to that DNA base. So it's going to continue to synthesize that RNA, single-stranded RNA piece, that what we call that transcript, that RNA copy of that DNA. until it hits a region that we call the terminator sequence. So there's, just like we have a promoter sequence that's embedded into the DNA, there's also going to be another region downstream, further downstream on that template strand. It's called the terminator sequence. Again, it's just a series of bases that RNA polymerase will recognize as this is the end. This is where I need to stop. And so it'll start its transcription at the promoter and end its transcription at the terminator sequence. Okay. So in eukaryotes, again, this is all still happening in the nucleus. And eukaryotes have this extra step that we don't see in prokaryotes. Eukaryotes, once RNA polymerase makes that RNA transcript, that single-stranded copy of DNA, eukaryotes are going to go through a process called RNA processing or RNA editing. And that's where pieces of that RNA transcript, so that RNA transcript, that original RNA transcript, which is a complete copy of that DNA gene, some pieces of this transcript will be taken out and some will be left in. So we're going to edit what's going to be left in and what's going to be taken out. It's called RNA processing. This happens in the nucleus. This happens this is done by a whole set of enzymes called the spliceosome, one of the coolest names of enzyme complexes, the spliceosome. So in eukaryotes, this only happens in eukaryotes. They're going to take out pieces that are what we call non-coding pieces. These are regions of the DNA that get transcribed into the RNA that do not contain information for how to build the specific protein. So these introns, they're intruders. They're not going to be used for that final product. So we're going to splice them out. So we're going to cut them out. We're going to splice them out. And then the exons that are the pieces that are going to be used to code for that specific protein, that contain the information for that specific protein, those are going to be spliced together. Those are going to be stuck together. Okay. So splicing just means basically cutting up that piece of DNA or RNA. Here we're cutting up RNA, and we're going to stitch back together those exons. So if we had one intron right here and one intron right here, the spliceosome would cut those out and then connect together these exons, exon 1, 2, 3. Okay, and then so only this region of only this region of RNA transcript is going to be used to build that protein. We're going to do some extra processing besides that as well. So that's the editing piece. Now we're going to add on a 5' G cap. So what that means that on the 5' end, we're going to put G's, just a few G's, just a few guanines. And then on the other end, we'll have a 3' what we call poly-A tail. We're going to put a lot of adenines on the other end of this transcript. So this 5' G cap, what it does is it's helpful in starting the process of translation. It's helpful in getting that RNA transcript to attach to the ribosome that's going to be responsible for translation. And the poly-A tail acts to help slow down the process of degradation, because these RNA transcripts, they're going to be hanging out in the cytoplasm. There's other enzymes that are going to be kind of bunching them down and making them shorter. And that poly-A tail kind of slows down that process of RNA degradation. Okay, so that's transcription. That's the process of making an RNA copy of DNA. And now this transcript can be used. It's going to exit the nucleus. It's going to enter into the cytoplasm, where it's going to find a ribosome, and it can be used to build proteins. So transcription, DNA is turned into a single-stranded RNA copy.